Hey guys, today we'll be talking about the PCR technique, or also known as the polymerase chain reaction. This was developed in the 1980s by a biochemist named Carey Mullis, who later on went on to win the Nobel Prize in 1993. This reaction basically takes a limited source of DNA at a crime scene or anything like that that could be a blood smear, it could be a hair follicle, or it could just be any kind of cell that contains some type of DNA, but it's very limited. And what this reaction does is it takes it in that small limited DNA, and it just amplifies it. It just makes a lot of copies to where you're able to do DNA fingerprinting, you're able to do DNA sequencing, and just you're able to do more when you have a bigger DNA strand. So I'm gonna take this camera and I'm gonna show you a couple drawings here. So what we have here, this is my drawing of a regular DNA, and sorry, I'm not an artist, but I'll use this pen to kind of guide it through. What we have here is the double helix DNA. Of course, it's 2D, not 3D. And this kind of turquoise color right here is going to be our targeted sequence, which can it can either be because that's all that you're able to gather at the crime scene, or just where you're trying to target or where you're trying to amplify. So for this reaction, you are wanting to have a sample that's within 50 to 2,000 nucleotides long. You don't want it any lower than 50, and you don't want it more than 2,000. It just won't run as smoothly. So from this small sequence, which like I said is between 50 to 2,000 nucleotides, within a few hours you can have millions, if not billions, of these targeted sequences. So we're going to move on to the first step of this cycle. So the first step is going to be denaturation. So what we do here, as you can see, you add heat in order to separate the two strands of DNA. So with this step, they're just wanting to kind of get that separated to kind of set them up for the following step, which number 2 would be annealing, which we hear these red little complementary DNA strands. They're synthetic DNA primers that are added in this reaction. They're short single-stranded DNA primers that are made from bacterium. Well, it was isolated from a bacterium named Thermus aquaticus, which is later known as TAC, and you'll see that in step 3. But what they do here is they cool off the strands after they're separated, because it was at 95 degrees Celsius, and now they cool it down to 50. This allows them to be able to add this synthetic DNA primer that they use in this reaction. So on to the next step. Oh, sorry, wrong piece of paper that I grabbed. The last step of this cycle is called the extension. The tag, as you can recall from the last step, kind of helps just finish up the targeted sequence complementary DNA, the synthetic one, so to say, and just helps it finish it off and just kind of get to a point where this can be broken off and get started over to the beginning of the cycle. So I'm going to switch back over to this right here, and I'll finish it off. After those are the 3 basic steps of just polymerase chain reaction, and with that, you basically do as many cycles as you need. Like I said, within a few hours you can get millions and billions, and the reason for this is there's a formula which is basically 2 to the n power. I don't know if that's the correct formula. That's the kind of formula I made up because it kind of makes sense, because 2, and you raise it to the number of cycles that you perform. So you start off with 2, or with 1 DNA, and then you break it off, which later makes 2 DNA strands. And then if you go to the second, another cycle, you add on, then those 2 will later split off and make 4. And then those 4 will split off and make 8, and so on and so on. So you can rapidly grow DNA in a short period of time. So that is basically all that I have for y'all. If you have any questions, any comments, just comment in this discussion. I do apologize. My setup was not the best. But if you have any questions about it too, that you might just want to read in the book, or if you just want to read more about it, it's on page 476 through 477. It's all in the top part of the page. And I hope this helped. Good luck, guys.